All right, welcome everybody to the next Single-Cell Omics Germany lecture. It's my absolute pleasure that we can host Gabriella Salinas today from Göttingen. And before we start with the actual talk, I would like to give you some background information. Our next lecture will be in the July 1st at 9:30 a.m., same time as today. It will be Dr. Tachibana from Munich. Talking about insights into the emergence of 3D genome architecture at the start of life by single-cell nucleus high-seq sequencing. Moreover, we are soon hosting a workshop. This is entitled Single-Cell Genomics Meets Data Sciences. The registration is not yet open, but will be open soon, so stay tuned. This is really a very nice opportunity to basically get insights into the world of single-cell genomics and its interfaces with data sciences. Then I would also like to quickly mention some points about our lectures. So we'll have a 30-minute presentation by Gabriela. This will be followed by a 15-minute discussion. If you have questions, please use the raise your hand feature at the end of the talk, or also type your questions into the chat box so I can read it out loud. This session will be also recorded, so in case you're interested in re-watching it or also watching past lecture series, you can basically access them in our YouTube channel. And yeah, with this, I would like to quickly introduce Gabriella Salinas to you. Gabriela studied biochemistry in Cordoba in Argentina. She then did her PhD in human genetics at the University Clinic in Hamburg-Eppendorf. After this, she basically joined the industry and became head of laboratory at the Novel Science International GmbH in Göttingen, where she established artificial intelligence-based drug discovery approaches. And in 2013, she joined the Fraunhofer Institute of Toxicology and Experimental Medicine in Hanover and coordinated projects for pharma companies in the field of toxicogenomics. And since 2007, she's head of the next generation sequencing integrative genomics core unit at the University Medical Center in Göttingen. And here her research interests include NGS-based technologies for genomics, transcriptomics, and epigenomics, and increasingly also with a focus on looking into single-cell genomic approaches. And this is also what she's going to talk about today. Her talk is entitled Technical Improvements in Single-Cell RNA Sequencing, Delivering the Promise of RNA Therapeutics. We're very much looking forward to your talk, Gabriela, and the stage is yours.
Thank you very much, Simon, for the kind introduction. And yeah, thank you for giving me the opportunity today to present my research and the research from my group. And I would like to talk today about technical improvements in single-cell RNA sequencing, delivering the promise of RNA therapeutics. And for this, I would like to start today with current integrated systems for single-cell sequencing, which includes systems that are able to dispense cells, proceed with the quality control of collected cells, as well as the downstream applications you can perform in the same instrument. And we are talking in general about RNA single-cell sequencing, but even I would like to present to you today other approaches can be performed, such as single-cell genomic DNA, or some other applications regarding epigenetics, such as ATEC single-cell. Afterwards, I would like to talk or to give you some improvement of a single-cell collection, documentation, and especially chemistry. Then to talk about the way you can perform transcriptomics within a cell in terms of evaluating messenger RNAs or poly-A transcript, but also at the same time to evaluate a large number of long non-coding RNAs. And finally, to talk about multi-omics data analysis, in this specific case, connecting genotype to phenotype within the same cell. So I think that screen is like, as you can see, in the panel A, you can see a microfluidic droplets system from the 10 genomics. And as you sure know, most of the people working in single-cell sequencing is using this platform. And more than 95% of the data reported in the Cell Atlas is coming from this platform. And it's, of course, the best platforms in terms of high throughput because you are able to evaluate several thousand of cells at once, especially helpfully when you are starting with very heterogeneous tissues. But we in our core unit realized that for many, not only clinical projects, this platform has limitations because most of the people needs, if you are interested on specific cells or subpopulation cells in starting with heterogeneous tissues, mostly you need to select cells based on different methods. But working with the 10 genomics, one of the methods is the fact sorting prior to sequencing or to preparation of libraries. Apart of this, most of the people are working with single nuclei because of cell sizing limitations, and all cells over 35 microns are mostly not accessed during the procedure cell capturing in the 10 genomics. Especially the chemistry the system is using, the 10 genomics, is they are using a oligo-DT for cDNA synthesis, and the cDNA synthesis is like partially. So you are able to sequence only short pieces nearly to the 3' or 5' of the transcripts, but the full-length approaches is still missing. For this reason, we introduce an additional platform which is well established as well as the 10 genomics. This is the isolate. The isolate platform is working basically using a nanowell plate. In this specific case, we are using a plate with 5,184 nanowells. And the instrument offers an integrated imaging system. Mostly you can work with a maximal of 2 fluorescence dyes. We are mostly using DAPI in order to distinguish living cells, and Texas Red to discard dead cells from the single cell suspension. Apart of this, the imaging system allows you to distinguish or to eliminate from the further processes cell debris or clustering or cell doublets and so on. The most beautiful thing for the isolate, I'm sure you all know this, is the flexibility in chemistry for RNA sequencing, because you can access transcript and very similar to the 10x, sequencing 3N fragments, but even you have the choice to go for full-length approaches, and the instrument allow you even to develop some self-made protocols or custom solutions that are not part of the, let's say, portfolio from Takara Chemistry. And what we are currently doing are only full-length approaches because of clinical questions. And we aim to evaluate a part of the transcriptomics. We would like to see how works the variant calling there, and or possibly to associate this with transcriptomic events. The disadvantage of the CellenONE is that the system, you started with 5,000 cells, but once we proceed with the quality control, only between 1,000 and 1,600 cells will be selected for further processing. And this is, of course, especially in comparison with the 10 genomics, too less. And for this reason, in panel C, you can see an additional instrument. This is the CellenONE instrument. It's basically a dispenser and that have the best and a really beautiful quality imaging system, because the system allows you to select cells based on morphology, independent of cell sizing, because you can access cells, whole cells, that vary in a range of sizing from 2 to over 500 micrometers. A part of this, we are using this in order to improve the capturing rate from 1,000 cells to 3,000 or 4,000, nearly 4,000 cells during one run. And for this, the first things we did in panel D is to combine the isolate with the CellenONE, using the CellenONE for cell dispensing and quality controls, using the Takara 5184 chip. This is the first things we implemented in our laboratory, and we are performing a quality control there. And once we dispense cells, we can use exactly the same chip and put this in the isolate for more for specific downstream applications. And today I would like to focus on RNA sequencing. For people that probably doesn't know how works the isolate is selecting by using a software from the company, is selecting living cells, and by staining and imaging, you can discard, for example, in panel B, you can see multiple cells, or in the middle, the dead cells, because the stain even in Texas red is visible, or cell debris, or cell debris, so that you can select the only good cells prior sequencing. Here, I would like to present you, this is shown by the first time in our group, because we currently are cooperating with people from the anatomy, from the group from Robert Friedrich. They are working with Schwann cells, and in this case, you can see peripheral nervous systems, cells that has a sizing, first of all, a very unusual morphology, and second, you will find cells between 200 and 500 micrometers in size. And by using 3 different fluorophores, we could visualize this in the isolates. And currently, we are able to access these difficult cells and sequence this in a whole cell manner without nuclei isolation. The next, so for this reason, we decide to combine the CellenONE system for cell dispensing based on cell morphology, based on cell sizing, or the instrument allow you to work from 1 to 4 different fluorophores in order to select real cells from heterogeneous tissues. And once we proceed with the quality control, we are working on the isolate then by using exactly the same chip. So the whole procedure from cell dispensing till library preparation occurs using one chip step. So in the isolate, the first things we are doing then after collection is proceed with the RT, pre-amplification, incorporation of indexing. Mostly this is the protocol for full-length approaches because we are performing only those. Then we proceed after indexing with the tagmentation reaction and the library. And this process from the cell dispensing till the library generation takes 2 days, fully automated. And most important thing is when we started with this novel system, we started this because people working, for example, in the cardiology, asked for the sequencing in whole cells mode for cells such as cardiomyocytes. And as you sure know, cardiomyocytes are very unstable cells, very unusual in morphology because the living cells and good cells are those with this cylindric morphology you can see in the middle. For this reason, we check how the instrument assesses cells over, let's say, 100 microns. And you can see in the first panel on the left, we started with a normal nuclei isolation after tissue dissociation, and the dispensing of cells is going very straightforward, as well as the library preparation. This is no problem, and you can collect cells within 30 minutes. In the middle one, you have an example of tissue dissociation enriched cardiomyocytes, and you can see different morphologies there. All of them are cardiomyocytes, but you can see that the other cells that are not cylindric is like apoptotic. It's not cell dead, but nearly to. And we could assess, based on this morphology, in the UMAP, you can see two different clusters that are coming from these different morphologies. And these kind of things you can also evaluate assessing quality control, and in order to perform qualities in morphology and evaluate the state of the cell. And in the last picture, this is an example from iPS cells differentiated to cardiomyocytes. These are cells with normal sizing from 24 to 50 micrometers, and the system is working really fine with this. In the next picture, we tested the performances of the single-cell RNA sequencing working with nuclei, and we have several oncological projects. In this specific case, we started or tested the system with frozen biopsies. In this specific case, what you can see is the evaluation of 3 different patients with pancreas cancer. And what you can see are 3 different phenotypes of pancreatic cancer. And this study has been done by using nuclei, because once you have frozen tissues, you are not able to work with whole cells because they are not living cells. And for this reason, we decided to match or to make a match study, starting with the original tumors from the patients. Then the researchers working in this project, they developed a PDX mice using the original tumor patient as well as organoids. And we performed with these different 3 patients a study demonstrating that nuclei is working really fine with the system. And if you take a look on the over part of the slide, the frozen tissues exhibit always less amount of exonic reads, which is normal, but what we see from the data analysis is that is still good for a gene expression analysis. So to put this in summary, the use of the CellenONE and isolating combination allow you to perform from the dispensing to the library preparation by using 1 step, only 1 chip, allow you the analysis of whole cells independent of sizing and cell morphology from 2 to 500 micrometers, and allow the analysis of living cells as well as fixed cells, which has been already tested by us in our laboratory, analysis of nuclei, starting with frozen tissues. And the most interesting thing is the flexibility in chemistry for different approaches of single-cell RNA sequencing. the 3-end approach, the full-length approach, improving the data analysis for detection variants, and transcriptomic analysis. And the last developed method by us was the single-cell total RNA-seq, which allow you a beautiful identification of long non-coding RNAs beside the messenger RNAs. Now we are coming to the second part of the presentation, because the first things we did in cooperation with the group from Andre Fischer from the German Center for Neurodegenerative Disease was to evaluate the performances of the total single-cell RNA sequencing on the isolate fully automated. And Andre is working on the on the identification of several long intergenic non-coding RNAs in human brain, a part of this astrocyte, microglia, neuron-specific studies, and even to find some long-coding RNAs in heart-specific long non-coding RNAs. For this reason, we started with him, and Andre is working since 2 or 3 years deeper with the 10 genomics and generate a lot of data. And the first things we did is to compare the performances of full-length approaches independent of single-cell total or the normal full-length approach. I have to say that all approaches are not done by using oligo-DT. Always we are talking about random hexamers in order for full coverage of transcriptomics. And what you can see is this is a pilot data specific for neuron-specific long non-coding RNAs data. So first of all, a global UMAP from the different kind of neuron-specific. And you can see that by evaluating isolate in full-length mode, the sensitivity of the assays in terms of detection of messenger RNAs or coding RNAs are higher than in the 10x, and especially if you are specifically looking at long non-coding RNAs, you can see that the isolate is the platform of choice. Now we are coming specifically because we are proposing a combination of instruments, CellenONE isolate, and for this, of course, we need to evaluate methodologically how good is the performances of the combination when we are comparing this with the isolate, which is, beside the 10x, a well-established single-cell system. And the goal was first to evaluate technically improvements by using this combination, and the second goal was to bioinformatically demonstrate how good you can assess calling variants that could probably be associated to transcriptomic markers in order to define phenotypically differences between samples. For this reason, we choose a special cohort of samples. In this case, we took progeria samples. These are dermal fibroblasts from patients, and we choose 6 different progeria samples that has been previously described as phenotypically different. And we include, of course, 2 controls, and started with the wet lab performances, in which we dispense exactly the same samples on one Takara isolate chip, 5,184 nanowells, and on the other hand, 3 different chips that should work only in the isolate as usual or as standard, let's say. Why this? Because in order to compare the methods, we need to generate after quality control about the same amount of cells. And you can see that in both, so merging data from 3 chips in the isolate, we got something like 3,100 cells, very comparable to the performance of the combination by using only 1 chip. In the table, you can see how many cells we dispensed from the very beginning and how much cells per sample we got after quality controls. I have to say that this quality control is not regarding the instrument itself, it's after bioinformatic analysis, evaluating housekeeping genes, performances, number of genes detected per cell in median, and whatever. So after the first bioinformatic data quality control. The first results we get technically, again, we started with exactly the same amount of cells for the isolate alone, as well as for the combination, which were about 3,000 cells. Then we sequence exactly in the same way to get about 400,000 reads in median per cell for both approaches. And the first things we observe is that by using the isolate alone, you will be able to assess much more exonic reads. But if you take a look on the figure A, the amount of intronic as well as intergenic reads from the combination is much higher. In the figure B, you can see that all transcripts we found in the isolate, in the well-established isolate, were found even in the combination, and much more, especially long non-coding RNAs, were found in the combination. And in figure C, you can see an RNA biotyping from both technologies. And even I show you in the slide before that the performances of isolate in terms of long coding RNAs in comparison with the TEDx is excellent. But you can see here that when you compare the combination, we are able to access much more long non-coding RNAs than working with the isolate alone. Okay, technically, we can say that the combination is working really fine, and that the overlap between well-established technologies, such as the isolate, is the performances of the combination is working really fine. Now we are coming much more in terms of biological evaluation. And the first things we did is, of course, this is a very common data analysis. It's nothing special, because we proceed with data visualization, how good cluster the patients in the UMAP from the combination and the isolate alone. In panel B, you can see a top 100 markers or genes found through the whole progeria samples, because in this disease, you will find many genes that are differentially expressed, that are common or associated directly to progeria syndrome. And in the panel C, we evaluate then in an unsupervised clustering, the general progeria transcriptomical pathways that we already know. For example, cluster 5 and 2 from figure C are much more related to the patient 13.9 and 13.60. Then you can see clearly cluster 4 and 3, which are much more associated with patients 615 and 800. And then we can see on the heat map some already known molecules involved in pathways are partially known from the progeria. And we evaluate, as usual, some kind of genes that has been described as a kind of markers for specific phenotypes under the UMAP. And once we evaluate transcriptional profilings, the next step was to perform a single, to determine variants, SNP calling variants within the samples. And for this, because we are looking at phenotypically differences, we determine the following conditions. First of all, because of single-cell RNA has a lot of noise, you have, in order to assess variants, to use additional data sets such as bulk RNA sequencing, whole exome sequencing, so approaches similar to those, in order to confirm all variants you are getting from the single-cell data. The second condition was, of course, to evaluate known synonymous mutations, because this is a very experimental bioinformatic data set. We checked that most of them were already reported in the database from single nucleotide polymorphisms. And because of phenotype description, we searched specifically for those variants that were known uniform distributed. This means patient-specific. And we got a list of several hundred of those variants for each of the samples. And you can see here a visualization of very few of them. And we tried, of course, to define some variant for each of the patients that are labeled in red in the UMAP. The next step was, based on these variants, we performed similar to one study from Petty 2021, published in Nature Communications. There was a linear regression tool in order to associate. I have to show you the regulomes in much more detail. We performed this kind of regulome analysis. On the left, you can see all 8 samples we analyzed. Regarding this specific mutation, in this case for patient 615, and mutation in the gene DAX at this specific position. And you can see the transcriptional effect regarding these variants. And those genes on the upper part of the regulon or heat map are transcriptional markers suggest to be direct related to this specific mutation. This is, of course, bioinformatically calculated, and non-functional study has been done till now. For this reason, we decide to proceed with the next step, which is in order to confirm the regulome results, we took each of the sample patients, in this case, sample 615 and sample 1360. We took the mutation we found for the gene DAX, as well as the mitochondrial CYP, each of them for each of these specific patients. And what we found, you can see on the left in red, I put all genes that has been commonly detected for this sample when we are doing differential expression analysis between mutated cells and non-mutated cells from this specific patient. And what was really nice to see is that when we are doing reactome and connecting all of these molecules, we were able to define some pathways specific for this specific sample based on this mutation, which was for sample 615, the transcriptional regulation of TP53, that confirmed results from transcriptomic I show you from the very beginning. A part of this, we realized that the gene DAX itself was differentially expressed, not only mutated, and is strongly associated with alternative lengthening of telomeres, which is the main pathway described phenotypically for these patients. On the other side, you have the other patient with the mitochondrial mutation, and the mitochondrial dysfunction in progeria is the second main pathway associated to progeria. And we could find lot of overlap of genes in red, even again, that were found transcriptionally as well as in the regulome, as well when we are doing differential expression analysis between mutated and non-mutated cells. In this specific case, we found when you are doing reactome, that's the transcription regulation of MSTC is regulated, as well as the MET activation, and MET activation is something that has been previously described, so that we could test by using confirmations of variants in single cells with other data sets, by using data previously published, that the tools bioinformatically we apply for this are reliable in this kind of thing. So lastly, so I would like to say that improvement for RNA therapeutics consists of the choose of downstream application of RNA-seq depending on the biological question, the processing of long non-coding RNAs, and you sure know that most of biomarkers currently defined for disease, especially in oncology, are long non-coding RNAs, to evaluate genetic variants associated to transcriptional markers, and to evaluate samples phenotype based on transcriptional and genetic events. So now I'm finished with this presentation, and of course would like to thank my group, which consists of technical, excellent technical assistants, as well as bioinformaticians working not only in service staff, much more in research, and students, and especially groups and institutions in Göttingen has been working in the development of single-cell platform, especially the German Center for Neurodegenerative Disease, Professor Andre Fischer, the cardiology, Professor Hasenfuss and Hubertus Zimmermann, the oncology and the human genetics by Professor Dr. Bernd Bollneck. Thank you for your attention, and so I'm happy to hear if you have some questions.
Yeah, thank you very much for this very nice presentation. Technically very interesting, also very interesting biology. If you have questions, please raise your hand, or also type your questions into the chat. That would be great. So I can already start with the first question that has been pasted into the chat. It comes from Rodrigo Coutinho de Almeida. And the question is, did you look for alternative splicing events in the ICEL8 in combination with the CellenONE?
I have to say that the evaluation of splicing variants by doing full length is of course possible, especially when you are doing the single total RNA, because the coverage is much better. But we never look specifically at this, much more how would is a coverage and how deep you need to sequence in order to perform calling variants with this. But in theory, the evaluation of this is possible.
Mm-hmm. Thank you very much. I personally also have a question. Until we wait, maybe there will be more questions, and I'll ask you in the meantime. What about the costs? I mean, I think you kind of presented very nice data on the advantages of combining like the CellenONE and the ICEL8. That looks very, very interesting, very promising, particularly for the kind of large cell types. Can you say something about costs in comparison to 10x Genomics or other platforms in terms of per cell costs and overall costs?
Yeah, exactly. So I already said that the 10 Genomics is evaluating about 8,000 cells at once. And if you ask about cost per cell, sure will be the 10 Genomics, probably cheaper. But what I can tell you is the cost you will need per experiment, and I'm talking about per chip. So evaluation of 3,000 to 4,000 cells. You have two different steps to consider. First of all, the dispensing RT chemistry doing libraries for one chip, which is about 3,900 euros or 4,000 euros around, and then the sequencing cost. And this depends, of course, what you would like to do with the data. In this particular case, in which we sequence deeper in order to determine variants, as well as deeper transcriptomics, long non-coding RNAs, whatever, we sequence 400,000 read per cell. And these are mostly two lanes in the high-sec 4,000, for example. And these are costs of about 2,500 only for sequencing. So then you need to add this to the 4,000 before, 6,500 euro per experiment with a very deep sequence.
Yeah, cool. Good to know. And if we already get like your direct feedback from a facility, how many users are at the moment kind of using, for example, this platform versus others in your lab?
Exactly. This is a good question because we are working very close to the human genetics. And we are working much more in clinical trials, so in clinical issues. And for this reason, it will be in the future essential to evaluate variants beside transcriptomics. And most of the researchers work in the cardiology that would like to assess whole cells and whatever. So we have 80% of the people using this technology because it's working fine. But we still have other people working with 10x. For example, Andre from the German Center for Neuro, he's still working with the 10x, but in case he's looking for long non-coding RNAs, he knows he has to generate data with the isolate platform much more than. Depends, always depends what you would like to do.
Sorry, I was on mute. So I ask you once more to paste questions or raise your hands if you have some. At the moment, I don't see raised hands. I ask one more questions in the meantime. So I was very intrigued by the long non-coding RNAs that you can detect now. I think it's very exciting and indeed, of course, very difficult with the classical approaches. So from a more biological term, I mean, if you basically do these types of analysis and only subset your data on long non-coding RNAs, do you also see cell type separation? So does basically every cell type has really a unique composition of long non-coding RNAs?
This is an excellent question we have to do. I have to say that we started with the single-cell total RNA a couple of months ago, mainly working with neurons. And what we are doing is UMAP from all of them. We consider not only long non-coding, even the poly-A transcripts. But sure, with the data we already have, we can take a look at that, but we never perform only for this.
Okay, yeah, very exciting. I'm very much looking forward to see how this looks like. I think this would be really cool.
Yeah.
Yeah, there's one more question coming in, which I think will be our last question, because then we're also kind of at the end of the time. So Ipek Göttingen asked you, hi, could you maybe explain the difference in the UMAP between the methods with iCell8 only versus iCell8 joined with the CellenONE?
So to think that by using, probably if we did this study with Untitled Genomics, you will get other UMAPs. So UMAPs are not identical because you are using different chemistry. Even in the comparison, we are not using exactly the same isolate chemistry because of volume differences. So we are working with 250 nanoliters and volume in the isolate and 200 only in the isolate, in the combination with 250 and in isolate only with 200. So there is a lot of differences in volume and performances, still the same chemistry, but you are not able to generate working with different platforms, same visualization of clusters. And this can vary, and this is an important thing because I would like to sensibilize people to go out from clusters and to take a look at the data in much more detail. Many people need to understand what's mean a cluster. Our cells are probably much more apoptotic than because markers even are difficult to define cell types mostly. In the single cell atlas, the information coming from different cell types and tissues is even very heterogeneous. And for this reason, I think that to show you maps and to over-interpret this based on cluster is always a mistake researchers are doing.
Thank you very much for this explanation. Very interesting. With this, we are slightly over time, and I would say I would like to thank you again, Gabriela, for giving this really nice overview, both on the technical aspect, and particularly, I think, important for fragile cells and for large cells. I think it's a really, really important contribution here and really important insights, but also in all the other aspects that you mentioned. Really interesting presentation. I would like to remind you that in case you missed something, and this session is recorded, so you can basically re-watch it if you're interested in specific details that you may have missed. I would also like to remind you that in 2 weeks, we have another SCOG meeting or SCOG presentation by Dr. Tachibana from Munich, the 1st of July, same time as today, 9:30. With this, yeah, thanks again, Gabriela, for this nice presentation, and I wish you all a great Friday and a nice weekend ahead of you.
Thank you, Simon, for your kind introduction and to be there. Thank you very much.
Thank you.